Hi, thank you for coming to my talk. I'd like to start by acknowledging my co-authors who helped me make this project possible. Today, I'll be presenting on our first results from a precipitation manipulation experiment that we started 2 years ago. Anthropogenic climate change is impacting every ecosystem on the planet with profound effects on biodiversity and ecosystem functioning. While temperatures are increasing globally, though at different rates in different places, predicted changes in precipitation are much more variable, with some places becoming drier, some becoming wetter, and with substantial changes in the frequency and severity of precipitation events. Understanding these changes to precipitation regimes is especially important in arid ecosystems. These systems, which include deserts and arid and semi-arid ecosystems, are found on every continent and make up 41% of the terrestrial landmass. Arid environments frequently exhibit high ambient daytime temperatures, sporadic precipitation, and spatially or temporally sparse nutrients. Importantly, species in these arid ecosystems have evolved different strategies for coping with these limiting resources. Some species are more plastic and can rapidly respond to predictable precipitation. Annual plants are a good example of this, where they quickly grow and set seed when the rain comes, and wait for a good year to do this again. Other species are bet hedgers and have a strategy to simply persist through the bad times. These species are often found in less predictable environments. Perennial shrubs are a good example of this, where they survive through the bad times, but are not able to respond as quickly to the good times. An additional challenge for understanding how altered precipitation may influence arid ecosystems are the seasonal dynamics to rainfall. Many arid ecosystems, such as parts of the southwestern United States, receive both winter and summer precipitation. In some cases, this winter rain is more plentiful and more predictable, while the summer rain is less plentiful and comes in highly variable monsoonal storms. Our overarching research question is to ask how biodiversity and ecosystem function respond to altered precipitation in an arid ecosystem with variable seasonal precipitation. We are conducting this research at the University of California, Riverside's Boyd Deep Canyon Natural Reserve, the blue star on the map on your left, which is located at the crossroads of the Sonoran Colorado Desert and the coastal Mediterranean ecosystems. The plant community here receives both winter and summer rainfall, resulting in distinct annual plant communities in each season. And there are a variety of perennial shrubs that persist through both seasons. The research I will be presenting on today is focused solely on the annual plant communities at this site. Precipitation at our research site is quite variable. The figure on the left is the seasonal precipitation totals for our research site from 1989 to 2019. The orange points are what I'll be referring to as the summer season, which goes from July through October to November. And the blue points are the winter season, which goes from October, November through June. Note that the seasons are of different length. The figure on the right is a response ratio of precipitation, where positive values, blue points, indicate that more of a given year's precipitation fell during the winter season, while negative values, orange points, indicate that more of a given year's precipitation fell during the summer season. Interestingly, from 1989 to 2003, the first 15 years of data, only 2 years had more summer than winter rain. While the more recent 15 years, 2004 to 2019, had 5 out of 15, or a third of the years, with more summer than winter precipitation. To answer our overarching questions, we set out to assess how biodiversity and ecosystem functioning were influenced by the precipitation in each season and reduced and increased precipitation in each season. To do this, we built the Pinion Flats rainfall manipulation experiment in spring of 2018. This experiment consists of 24 about 52 meter square plots with the following treatments arranged from overall driest to wettest. The colors here will be used in all the results figures throughout this talk. A summer-only treatment, in dark orange, received ambient summer precipitation and had a roof during the winter to exclude winter rainfall. The winter-only, light orange, received ambient winter precipitation and had a roof during the summer to exclude summer rainfall. The control in green received both ambient summer and ambient winter precipitation. The summer plus in light blue received ambient winter precipitation and had water added during the summer. And finally, the winter plus, dark blue, received ambient summer precipitation and had water added during the winter. On the summer only and winter only plots, we erected metal frames and covered them with polyethylene plastic. These were aligned with the prevailing winds to help with airflow under the roofs. Summer only roofs were placed in early summer, July 7th and 23rd of 2018 and 19 respectively, after the winter plants had senesced. And they were kept on until early fall in November and October, after the summer plants had senesced, when we moved those roofs to the winter only treatments. During each season, precipitation was collected on the roofs by way of a PVC system that runs the 4 1,500-gallon tanks down slopes of the roofs. In the summer, we added the water that fell on the winter-only plots to the summer plus plots. And in winter, we added the water that fell on the summer-only plots to the winter plus plots. By pumping water from the storage tanks up to the plots and distributing it evenly across the plots using 17 millimeter Netafim drip tubing, with water meters at each plot to ensure the same amount was added to every plot. Over the course of the experiment, we added water to the water addition plots within a week of a given precipitation event. This figure has the monthly precipitation or precipitation equivalent received by each treatment on the y-axis and each month of the experiment on the x-axis. As you can see, the winter plus plots received more water than the summer plus plots, as there was more water to add in the winter. Moreover, the amount of water added was variable and depended on the previous precipitation events. In total, the summer 2018 plus plots received the equivalent of 29.2 millimeters of additional precipitation. The 2019 winter plus plots received the equivalent of 132.9 millimeters of additional precipitation. The 2019 summer plus plots received 21.6 millimeters, and the 2020 winter plus plots received 107.8 millimeters of additional precipitation. The schematic on your left shows our plot setup for each of the 24 plots. Each plot was divided in half, with one half set aside for manipulative experiments and one half used for the monitoring data I'll be presenting on today. In each plot, we established 6 meter squared subplots, and in the center half meter square, the light blue squares, we collected data on species composition for the annual plant community using point intercept approach, recording all hits at 50 locations. In the green quarter meter square, we harvested all above ground biomass at peak biomass in each season. We rotated the location to not resample biomass in the same place. The picture on the right shows a water addition plot in the foreground and a rainfall exclusion plot in the background. So even though there was more water added in the winter, as you saw in that previous figure, the winter annual plant community showed no biomass response to additional precipitation. This figure has biomass in grams per meter squared on the y-axis, and all 4 figures have the treatments on the x-axis. From left to right, we have summer 2018, winter 2019, summer 2019, and winter 2020. In summer 2018, the winter-only summer exclusion had significantly lower biomass than the control, the brown arrow. And the summer plus had significantly greater biomass than the control, the green arrow. And we found the same pattern in summer 2019. In winter 2019 and 2020, we found that the summer only, the winter exclusion treatments, had significantly less biomass than control treatments, and the winter plus did not differ from control. These patterns suggest that if climate change results in reduced precipitation in any season, it will have negative effects on biomass in this ecosystem. Whereas if climate change results in increasing precipitation, it will only increase biomass in the summer and not in the winter. While we saw an impact of increased precipitation on biomass, we found no effect of increased precipitation in species richness or Simpson's diversity. Here we have the same layout as a previous graph, but for the top row, the y-axis is mean species richness per plot, and the bottom row is Simpson's diversity. In 3 seasons, summer 2018, winter 2019, and winter 2020, we found that reducing precipitation negatively impacted both species richness and diversity, the brown arrows. But in no case did increasing precipitation affect either richness or diversity. Moreover, we found no effects of any of our treatments on richness or diversity in summer 2019. These results suggest that decreased precipitation in any season will negatively impact biodiversity. There may, there may be stronger impacts in winter than in summer, as the reductions were greater in winter, and one summer season had no response. Interestingly, increased precipitation does not increase richness or diversity. Although altered precipitation has modest impacts on richness and diversity, we did find significant shifts in community composition. Here's a non-metric multidimensional scaling plot of Bray-Curtis dissimilarity among plots. Each point represents the mean axis scores for the treatment, and the bars represent one standard deviation. We're using the same color scheme as in the previous figures, but now the shapes represent the different sampling periods. First, we found that community composition differed among seasons. All of the following p-values are based on permanovas with 9,999 permutations. Here you can see that summer annual plant community with the orange circles differed from the winter communities in blue. And the summer community was dominated by Portulaca and Bouteloua, while the winter communities were dominated by Erodium and Bromus madridensis. Within all 4 sampling periods, we found that our treatments significantly differed in composition, but these differences varied by season. In the next 4 figures, I'll be highlighting each sampling period's treatment differences and including indicator species on the left side of the figure. In summer 2018, we found that the significant differences in community composition among treatments were primarily driven by the species in the winter only and winter plus treatments, with no difference between control and summer plus treatments. In winter 2019, we found that the significant differences in community composition among treatments were driven by species in the winter only and summer only treatments, and that the winter plus treatment did not differ from control. In summer 2019, we found that the significant differences in community composition among treatments were driven by species in the winter only, and summer plus treatments. And this was the only instance where the summer plus treatment significantly differed from the control. And this difference was driven by the native annual grass, Bouteloua aristatoides. And finally, we found that the significant differences in community composition among treatments in winter 2020 were driven by the summer-only plots, with all other plots having very similar composition, and the winter plus treatment did not differ from control. In summary, we found that decreased precipitation had negative impacts on biomass in all seasons, negative impacts on richness and diversity, with stronger impacts in winter, and resulted in shifts in community composition in every season. In contrast, increased precipitation had a positive impact on biomass in the summer, but not in the winter, no impacts on richness and diversity, and no shifts in composition in winter, but a shift in composition in summer after 2 years of increased precipitation. Taken together, our results suggest that altered precipitation in these seasonal arid ecosystems is nuanced, and that increasing and decreasing precipitation won't simply have opposite effects. While decreased precipitation tends to have negative effects, increased precipitation can have positive effects, but it really depends on the season. For this ecosystem, increased summer precipitation will likely increase biomass and lead to a shift in species composition in the short term. And these changes may lead to more drastic changes in biodiversity and functioning in the long term. We plan to continue this experiment for several years, and in the short term, we plan to begin quantifying functional diversity and to experimentally assess individual species responses to altered precipitation. With that, I'd like to thank the many people that helped on this project and the Boyd Deep Canyon NRS for logistics support. And I would especially like to thank Lisa Schauer, Shane McFall, and Miguel Solis for their long hours making their project work. Thank you for listening, and I'll be happy to answer any questions.